Hello everyone and welcome to Monica's Worm Corner. On this segment we're going to be talking about RNA interference, also known as RNAi. And so RNAi is used by many organisms in order to regulate gene expression. It's also used by scientists in order to identify the function of a gene or to identify potential genetic therapeutics that can either prevent a disease or help kind of delay the progression of a disease. And so what I have drawn on the left-hand side of the board is the central dogma, and so the step of the central dogma impacted by RNAi is the RNA step. So what ends up happening is RNA is silenced, and so that protein is not produced. And so in our lab, what we do is we feed our worms double-stranded RNA. So once that double-stranded RNA enters the cytoplasm, it's going to be cleaved by Dicer, and so Dicer cleaves it into these small fragments that are 20 base pairs, and they're called small interfering RNA, also known as siRNA. So another small RNA is also microRNA, known as miRNA. And so microRNAs are when mRNA is folded and processed before it is exported out of the cytoplasm. And so for the steps of RNAi is that the siRNA is going to bind to an argonate protein, and then the siRNA, the only part of the siRNA that's going to stay on to the argonate protein is the guide strand. And so this combination of your argonate protein, your guide strand, and other proteins create a complex known as the RNA-induced silencing complex, which is called RISC for short. And so RISC with the guidance of the guide strand is going to then complementary base pair onto the mRNA target sequence, and so that complementary base pairing is like almost perfect, and so it's very precise. And so when they when RISC matches onto the target mRNA sequence, the sequence is going to be cut and degraded, and so that way your protein is not going to be produced. And so miRNA also guides RISC onto the target mRNA sequence, but it's much less precise than siRNAs. And so with microRNAs, because of that imprecision, microRNAs can target hundreds of endogenous mRNAs. And so that is a little more insight into how RNA interference works. I hope you enjoyed this segment. Thanks so much for watching.